credentials to get CME and just highlight upcoming speakers. We have a visiting speaker from Montreal next week. The younger folks have requested Halloween no meeting, so we were responsive. And then we have Rick Nishimura from Mayo joining us in a few weeks after that, a fellows talk, and then Dr. Kirkpatrick. So a wonderful lineup of fantastic speakers coming up in the next few weeks. This is just our disclosure and accreditation. And with that, I'm going to ask Fadi to come up and introduce our speakers.
All right. So, welcome, everyone. It's really my distinct pleasure to introduce our speaker today, Dr. Al George. Dr. George is the Alfred Newton Richards Professor and Chair of the Department of Pharmacology and the Director of Pharmacogenomics at Northwestern University. I've known him for many years now, first by reputation, as I told Eric, as a luminary, really in the field of inherited arrhythmias, and subsequently as a colleague and friend. We served together as study section members on an NIH study section that he went on to chair. A bit of background on Dr. George: he received his MD degree in 1982 from the University of Rochester. He then went on to do his residency training in internal medicine at Vanderbilt and completed his clinical fellowship at the University of Pennsylvania. He then focused on research. He did 2 postdoctoral research fellowships that were focused on molecular genetics of ion channels, initially at the Institut Suisse de Recherche in Lausanne, and subsequently at the University of Pennsylvania. He joined the faculty at Vanderbilt in 1992, and enlightening speed within only 6 short years, he rose through the ranks to full professor. There at Vanderbilt, he founded the Division of Genetic Medicine and served as its chief until he left in 2014 to take on another very exciting challenge in Chicago as chair of the Department of Pharmacology at Northwestern, which I have to say he grew by over 3-fold since arriving there. It would take me a long time to list all of Dr. George's honors, and so I won't do that, but I'm just going to highlight a brief few. Dr. George has been elected as a member of the American Society of Clinical Investigation, the Association of American Physicians, and the American Association for the Advancement of Sciences. He's been awarded the Lucille Marquis Scholar Award in Biomedical Sciences and the American Heart Association's Established Investigator Award, as well as the Javits Neuroscience Investigator Award, among many others. Dr. George has really been a pioneer in the study of the structure, function, and molecular genetics of ion channels, both in the heart as well as in the brain. In fact, over the years, his laboratory has made numerous groundbreaking discoveries in this space. And I'm just gonna name one example. His work has led to the recognition of a unique form of long QT syndrome that arises from mutations not in ion channels themselves, but rather in calcium regulatory proteins, or protein called calmodulin, and this led him to coin the term calmodulopathy to describe a highly malignant arrhythmic disorder that really primarily affects the young, and I suspect we'll hear a little bit about that today. And so without further delay, Al, we're delighted to have you with us today, and I very much look forward to your talk.
Well, thank you very much, Fadi. And it's really an honor to be here. And I've really enjoyed meeting everyone that I had the opportunity to meet with today. Can everyone hear me okay? It's good in the back? Okay, wonderful. Let me just make sure I can advance the slides. So and I think I'm going to hire Fadi as my PR manager. But aside from that, it's a pleasure to tell you about this interesting story today of a novel genetic arrhythmia predisposition disorder that has been named calmodulopathy. And just as an outline of my talk, and these are somewhat more of the learning objectives, is I'm going to tell you about the clinical features of this disease and its genetics. We'll move to the pathophysiology and about its mechanisms that we understand to date. And that'll include some new data from 2 mouse models that we recently published. And then briefly contrast calmodulopathy with other similar cardiac arrhythmia susceptibility syndromes and to contrast them. So this is a cardiology grand rounds. I am an MD, and I have to start with a clinical case. So here is what got us into this discovery. This was a clinical case that was communicated to me by dear colleagues in Milan and Pavia, Italy, that being Leah Crotty and Peter Schwartz, who I worked with for many years. And they called me one day and said, we have this very sick infant in our ICU who has been having repeated episodes of cardiac arrest, ventricular fibrillation predominantly, and we cannot find the cause. The baby had a very long, prolonged QT interval. So they, of course, pivoted to doing genetic testing for the known genes associated with that syndrome and found nothing. The parents were completely healthy. So the assumption here was this was a child probably born with a de novo mutation or possibly some kind of recessive variant that was not evident on the genetic testing. But either way, we're probably dealing with a novel genetic form. And as that conversation was proceeding over a couple of weeks, a pediatric cardiologist, in Chicago, Bettina Cuneo calls me out of the blue and said, can I talk to you about a case? And here was another family with an almost identical scenario, even more severe. This was a young Hispanic woman, a child who was born to healthy parents, and they already knew in utero something wasn't right. The heart rate was slower than normal, which sometimes is a clue to a cardiac arrhythmia, a genetic cardiac arrhythmia. After she was born, her QT interval was measured and it was prolonged by over 700 milliseconds. She also had 2-to-1 AV block and evidence of electrical instability with T-wave alternans. And so again, because of the long QT interval, they pivoted to genetic testing for the known causative genes of that syndrome and found nothing. So this was around the time, fortunately, that I had received an NIH shared instrument grant to buy a next generation sequencer. And once that was installed, we started doing things like exome sequencing. So I suggested we should do exome sequencing on these two cases, with the assumption is that they have de novo mutations. In other words, mutations that would not be clearly present in the germline of the parents, but would be present in the offspring who are affected. And so this is the paradigm. We sequenced the trio, parents and child, and then we looked for the overlap of variants between the parents and the child. And we looked for variants that were not in either parent, but were present in the child as potential candidates for a causative gene. And so the way the analysis went, we first looked at how many total variants were in each sample and each proband. We then subtracted from that list all of the parents' variants. So we're left with 36 or so variants that we deemed de novo. And then with a series of other filtering steps to make sure we could identify protein altering variants, novel variants that weren't in the general population. And then this was early days of next generation sequencing, and I guess we didn't quite trust it. So we went and did Sanger sequencing, the traditional way of doing DNA sequencing. And in the Italian proband, there was only one variant that survived that. And that was a variant in a gene I had never heard of, with the abbreviation CALM1 or CALM1. And I was anything but calm at that point because the second case had two variants, one of which was in a gene that wasn't expressed in the heart. So we discounted that. And then the other gene was another gene that encodes for calmodulin. And so why two different genes coding for calmodulin? Well, I'll tell you in a moment, the real story. There are three genes that code for calmodulin. So what is calmodulin? Calmodulin is a ubiquitous calcium signaling protein. Its structure is quite simple. It has these two helix turn helix motifs that each bind two calcium ions in a motif that's referred to as an EF hand, and this is sort of the inspiration for that. And by binding calcium, it then undergoes conformational changes that allows it to interact with different effectors, including some ion channels, some enzymes, and other proteins in tissues. And calmodulin, by the way, is everywhere. It's in virtually all tissues, a ubiquitous calcium sensing protein. This dichotomy of the two genes really threw us off there for a while, but then it turns out that this was well known. There are 3 genes dispersed in the human genome that code for the exact calmodulin protein, identical at the protein level, different enough at the nucleotide level that you could distinguish them. So here we've got 3 genes, 2 of which we've now just implicated in a severe, life-threatening congenital arrhythmia disease. So what does calmodulin do? I mentioned briefly, it binds calcium. It then interacts differentially with different targets in the cell, too many to list on one slide, but 2 of them, which are important to understand this disease, are the L-type calcium channel in the heart and the intracellular calcium release channel, or ryanodine receptor. And those 2 will come up later when I talk about pathophysiology. So back to the genetics. So we had found these 2 cases. These were lone cases of de novo mutations. And clearly we wanted to know, was this common? Did we just miss this gene earlier in cases of long QT syndrome, for example, for which genes weren't found? So again, with my colleagues in Italy, they identified 82 mutation negative, meaning the mutation wasn't identified yet. And we screened them for mutations in all 3 calmodulins. And from that work, we found additional cases. We found 2 more cases, one of which was, all of which were de novo. And all of these folks shared these features. They had cardiac arrest during infancy. They had ventricular fibrillation, sometimes preceded by an arrhythmia that looked a little like torsade de pointe, which is the classic arrhythmia of long QT syndrome, but not always. All had very long QT intervals, frequent T-wave alternans. Some of them had 2 to 1 AV block. And a few of the cases had neurological phenotypes, which got my attention. So were these mutations, how are they affecting calmodulin functionally? So to that, I turned to a biochemical colleague, Walter Chazen, and his postdoc, Chris Johnson, who had a way of purifying calmodulin and measuring in vitro the calcium binding affinity of wild type calmodulin and the mutations. And so when we did that for these 3 novel mutations that we discovered, there's actually 4 mutations, but the amino acid is so conserved that 2 of them were essentially the same amino acid substitution. So we studied the 3 novel amino acid substitutions. And what we found was in the C lobe, or the carboxy tail of calmodulin, the 2 EF hands that are present, EF hand 3 and 4, the mutations were either in residues, acidic residues, aspartic acids or otherwise, that were important for chelating calcium, or were nearby that potentially could disrupt the folding of the EF hand. And in all 3 cases, they discovered that compared to wild type calmodulin, the mutant calmodulins all had lower calcium binding affinity. So at a molecular level, this was a loss of function mutation. Over time and subsequent years, and our group and many other groups began to screen for calmodulins, and it ultimately has become institutionalized on genetic testing panels. So there are more and more cases being discovered. And this is sort of a summary of a few years ago of what we knew. And so all 3 genes have been now implicated in some form of inherited or congenital genetic arrhythmia. And the phenotype is variable. It can either present as if it were long QT syndrome, like the cases I presented. It can also present as a catecholaminergic induced arrhythmia, like CPVT, or just idiopathic ventricular fibrillation or sudden unexplained death. And there's a lot of overlap between these. And when we've looked at the calcium binding affinity of the majority of these, they're all abnormal, with the exception of a few limited to this one particular variant, which was a CPVT variant. But most of them have impaired calcium binding affinity. Now, there's been a worldwide effort to collect these cases, again, led by my Italian colleagues, and they've published recently the results of a years-long collection, which they've referred to as the International Calmodulin Registry. And so this is sort of bringing you up to date. This was just published a few months ago. They've ascertained 140 individuals worldwide that had this condition. There was a total of 97 individual families. It's about equal male-female difference. There doesn't seem to be a sex bias here. And then among those individuals, there was 60-some unique calmodulin variants. And here's how they distribute among the 3 genes. Now, in the ClinVar database, which banks data from genetic testing companies, I found just a few days ago that there's 150 different missense variants. So I think this registry is close, but it's not capturing everybody. And I think, again, this is something that I think will emerge as more testing gets done. Now, what's also interesting is, although I presented these 2 cases as emblematic of how we were drawn to these families and found calmodulin with severe cardiac arrest during infancy, that's probably not as severe. That's probably more severe than the average. Turns out the average age of the first event is probably not during early infancy. It's probably later, a little later in childhood. And first events can be as late as mid-teenager, late teenager years. Most are symptomatic. There are a few families where the calmodulin mutant has segregated from generation to generation, and some of the parents and siblings are not affected, at least overtly. Most of them do have some arrhythmias or cardiac arrest, and sadly sudden death in some. Then the phenotype is interesting. Again, many of them have a longer QT interval than normal, so we would call that long QT syndrome. Others present with an exercise-induced arrhythmia, which we would think more like CPVT, but there's a lot of overlap between those 2 syndromes, so it's not exclusively one or the other. And there are some atypical cases. I'm going to finish my talk later today with a particularly unusual case that I think is worth knowing about. And this relates a little bit to the fact that in those first series, we saw neurological symptoms, but really weren't sure if those were the byproduct of a primary defect, of the calmodulin mutation, or was this a byproduct of having cardiac arrest and central nervous system anoxia? And so in this registry, they found that probably there was a small subset that had neurological symptoms, either seizures, developmental delay, possibly autism rarely, that were not related to having cardiac arrest. And then lastly, there were some cases with structural heart disease that were also noticed in the workup. Most frequently, it's left ventricular non-compaction, which is not specific to this syndrome, of course. Now we moved on to try to understand what was the pathophysiology of these conditions. We knew calmodulin was important. We knew many targets that it could affect, but proof was missing as to what the mutations did. And so through a important collaboration with the late David Yu, then at Hopkins, we studied the mutations in a cardiomyocyte model. These were guinea pig ventricular cardiomyocytes that were then transduced with an adenovirus carrying either wild type calmodulin or one of the 3 mutants. And now, initially, this study only focused on the L-type calcium channel. And if you depolarize a cardiomyocyte in the presence of extracellular calcium, calcium channels open, and then they partially inactivate. If you substitute barium ions for calcium, and calcium channels conduct barium ions very well, you see the same degree of opening, but you do not have this inactivation. So the difference between this time course of inactivation between conditions having calcium as the charge carrier or barium is referred to as calcium-dependent inactivation, just a simple term, or abbreviated CDI. So this is what happens if you overexpress wild-type calmodulin. What happens if you put the mutants in? You completely abolish calcium-dependent inactivation. So what this is a molecular loss of function. Calcium affinity is lower. These are calmodulins that have impaired calcium binding. But in the context of a cardiomyocyte, you see a gain of function in the calcium channel. They don't inactivate. You could call that loss of inactivation. But if you're looking at the net effect of this, the net effect is more calcium current, which will affect the action potential plateau. And so to prove that, current clamp was done in those same cells, And here are the action potentials recorded from those cells, paste action potentials, with wild type overexpressed calmodulin. And here is one of the mutations. They created this very bizarre long action potential that interrupts the next beat. So this shows the substrate for what would be long QT syndrome, as well as AV block, 2-to-1 AV block. That was present both in this and in one of the other mutations. There's some oscillation there, some alternating action potential duration as well. And what's fundamental here is that these are defects we could directly trace to impaired calcium-dependent inactivation of the L-type calcium channel. There were mutations that were associated with either a pure CPVT-like phenotype with normal QT intervals, or with a mixed picture of a long QT and a CPVT-like picture. Those mutations behave a little differently. So here is the pure CPVT mutation. And this is the one I pointed out earlier that has normal calcium affinity. And this is a mutation that's outside of those calcium binding EF hands. This mutation does not affect calcium-dependent inactivation at all. And here's a second mutation, N98S, which turns out to be the most recurrent mutation in this gene family. And here we get a partial impairment of calcium-dependent inactivation. At the action potential level, the CPVT only variant doesn't affect action potential duration, as we could have predicted, but the N98S prolongs action potential. So this is beginning to fit that impaired calcium-dependent inactivation is associated with the long QT interval. Now, those CPVT mutations, or those that have an overlap, were the subject of a second study that was done in collaboration with Bjorn Nolman and Don Burrs and others, where this was a different model system. Here, mouse ventricular myocytes were permeabilized with a detergent that will allow extracellular proteins, including calmodulin, as well as the extracellular calcium, to equilibrate with the intracellular components of the cell. And so this is one way to introduce calmodulin mutants into these myocytes. And then the readout was a fluorescence-based calcium recording. So here is essentially calcium waves recorded in the myocytes. These are spontaneous beats. And here's what happens when you overexpress the CPVT mutation. You get a faster generation of calcium waves. And you saw the same thing to some slightly lesser extent with this overlap mutation N98S, which again causes both a long QT-like phenotype and a CPVT-like phenotype. And this was reminiscent in Bjorn's lab of what you see when you look at a pure CPVT mouse mutant, which is a homozygous mutation of knockout of calsequestrin. So this suggested that there was a defect in intracellular calcium release, presumably mediated by the ryanodine receptor. Now, in these cases, if we looked at the mutations that were associated with a more pure long QT syndrome, you got pretty much normal or slightly slower calcium waves. So this was a mechanistically distinct way. To prove this was a ryanodine receptor mediated event, Derek Lever purified ryanodine receptors and then exposed them to either wild type calmodulin or some of these mutant calmodulins. And in this case, this is basically single channel recording in a bilayer of individual ryanodine receptors in the presence of low calcium and wild type or endogenous calmodulin up to this point where the mutant calmodulin was introduced and the open probability jumps dramatically. So this is just a summary of the data. This is the open probability. The height of these bars indicates how much time the ryanodine receptor is open in conducting calcium out of the sarcoplasmic reticulum. And in both cases, the N54I and the N98S, the two CPVT-like mutations, had increased open probability, which again is a known predisposition at the cellular level for cardiac arrhythmia. If you lowered the calcium, the one CPVT mutant still had increased open probability, but not the one that had the dual phenotype, and still haven't got our head around why that is. Now, the other interesting thing about this condition is, as I said a few minutes ago, there are 3 calmodulin genes, but all the mutations we found, the carriers were heterozygous. So there's a total of 6 copies of calmodulin, encoding calmodulin, and only one of the 6 had a mutation in it. So there must be some dominance of that mutation allele. So in this case, going back to the mouse permeabilized mouse ventricular myocyte model, here different ratios of the wild type and the mutant calmodulin protein was equilibrated with the myocyte. And here with pure mutation, we saw this increase in frequency of calcium waves. And if you went to a 50-50 ratio, still had increased frequency. And even at I think this is 1 to, what was that? 1 to 8 or so, you got still an increase. So this suggested that there was a dominant effect of the mutation. Is that a dominant negative effect or something else? It's probably semantics. So these are both important models, the guinea pig model and the mouse permeabilized model, but none of them are human myocytes. And we turned next to looking at how these calmodulin mutations would impact the dynamics of these events electrophysiologically in a human myocyte. Now we got human cardiomyocytes from iPS cell technology, and we applied that first in a novel mutation that we discovered at Northwestern with help of Greg Webster, a pediatric electrophysiologist at Lurie Children's Hospital. And this was a case of a 3-year-old boy who had a sudden cardiac arrest, survived, and when he awoke, his QT interval was prolonged. He was treated with beta blockers and an ICD. The beta blockers didn't really work very well. He had another cardiac arrest later. Genetic testing found a novel calmodulin mutation, and that mutation was present in one of these important calcium binding residues in EFN4. And as we showed before, biochemically, this impaired calcium binding affinity. And we wanted to know, what if we put that mutation directly into a human cardiomyocyte? Now, at this point, we did not have iPS cells from the patient. We only had healthy control iPS cells from which we made cardiomyocytes. And so we simply transfected, using a lipid-based transfection system, a plasmid that encoded the calmodulin, either wild type or mutant, coupled with a green fluorescent protein into myocytes. And then we patch clamped the green myocytes. And we measured calcium current, which for a lab that prides itself on measuring sodium currents and potassium currents was a departure. Let me just say that. But anyway, calcium currents in these cells transfected with wild type calmodulin look normal calcium dependent inactivation, but the mutant transfected cells had lost calcium dependent inactivation. So this was a model that we embraced to look at a number of mutations to sort of bring out this molecular defect that was present. Now, a little bit more about the genetics, because again, these appeared to be mostly, in most families, de novo mutations. De novo meaning that they arose somehow spontaneously, either before or after fertilization in the proband, but were not carried in the full germline of the parents. But there is an exception to that, and that's called mosaicism, where individuals may harbor 2 genomes, one the predominant genome and one at a lower frequency. And that can vary by tissue, but it can also be present in the germ cells, either oocytes or in sperm. And so we actually, again, stumbled onto some cases where it looked like there was mosaicism, and to think about that for a moment. So this was the first case. This was a case submitted to us, suggested that we investigate from a family in Spain. This was a young girl who was born and had immediately a slow heart rate, which was due to 2-to-1 AV block, severe prolonged QT interval, T-wave alternans. Same story we've been seeing in these severe infants. Unfortunately, she had cardiac arrest, was resuscitated, put on beta blockers, had an ICD. Unfortunately, died at age 5 years, but not because of an arrhythmia, because she had severe hypoglycemia, which was attributed to a side effect of propranolol, which he was treated with pretty high doses. So not a cardiac death. The mutation in that case was novel. It was in the CALM3 gene. This was the first one we had found in CALM3. And it was another mutation that disrupted an important calcium chelating residue, in this case, a glutamic acid, changed to a positively charged lysine. And again, with the help of biochemical colleagues, Walter Chazen and Chris Johnson, we measured the calcium affinity, and it turns out that the calcium affinity, like the other mutations, was grossly impaired, about a multifold impairment. But that impairment was only in the C-terminal end of the calmodulin. The N-terminal end was okay. So this seems to, again, be a disease of loss of function of calcium binding to the C lobe of calmodulin. And we, again, turn to our iPS cell transfection model to show that the presence of this mutation disturbed calcium inactivation and prolonged action potentials. So again, using this as an example of two things. First, that we can, again, investigate a novel mutation. Where's the mosaicism? Well, we go back to the family, and this is the pedigree. Parents were healthy with normal electrocardiographs, and here is the proband who had this severe neonatal long QT syndrome, who later died, probably as a medication side effect. But looking at the family, they had multiple intrauterine spontaneous abortions. One of them had known to have fetal bradycardia, but unfortunately, none of these cases had DNA available. The two living siblings were healthy with normal QT intervals, but again, we didn't have the option of having DNA. Well, in the proband, this proband was heterozygous for the mutation. Interestingly, the mother was also heterozygous, but the mutant allele was only 25% of the wild type allele in her blood. And so this suggests that she was mosaic and perhaps presumably germline mosaic, so she passed it on to her child. Now, a more convincing example came to us via a geneticist in Switzerland who communicated to us this family from Saudi Arabia, who was a consanguineous marriage, and they had a series of children who died during infancy. A very tragic story. The two probands that came to light for genetic testing are shown here. Both had neonatal or infantile onset long QT syndrome, and by sequencing, they both had calmodulin 3 mutations that were deleterious, and all died. And there were some other cases that died without having genetic testing, but presumptively, this was, presumably they had inherited as well. But the parents were healthy, and the question is, was one of them mosaic? Now, a quick genetic screen of the parents showed nothing, but it seems improbable to have this many de novo mutations in a family if they weren't, one of the parents wasn't mosaic. So we did next generation sequencing, and in the father, we found that in the peripheral blood mononuclear cells, white blood cells in the blood, there was about 3% of the mutation allele that we could find by next generation sequencing. So we confirmed mosaicism, but it certainly didn't really jive with the fact that there were 5 out of 6 siblings presumably carrying this mutation. So it took a while, but we eventually got a sperm sample from the father, and the sperm showed 30% mosaicism, which is more consistent with this prevalence of the disease. I bring that up because we hear about, you may hear about mosaicism in different contexts, but we usually make that determination based on peripheral blood, which is just one of many tissues. What the tissue that matters are the germ cells. Did they pass it on from sperm or oocyte? Now, sperm is easier to get, clearly. But this is just important to know that mosaicism and the proportion of degree of mosaicism can vary widely among tissues. And so that's the rule of that. Now, I've said a lot about the genotype-phenotype correlation with regard to, on one end of the spectrum, are mutations that cause a more pure, long QT-like picture. And on the other end of the spectrum, a few mutations, not very many, cause a more pure CPVT-like pictures. And we think these have different mechanisms underlying them. But it turns out the most recurrent mutation, which occurs in CALM1 and CALM2, and so far not being seen in CALM3 yet, has been identified in now 22 patients worldwide, as according to the registry. All de novo. None of this is transmitted. All these people are unrelated. And so, at the time, we were also, this was years ago, we were aware of 6 or 7 cases of this mutation that were from various places. And there was 3 in the CALM1 gene at the time and 4 in the CALM2 gene, all the same amino acid substitution. And they were from different places in the world and different backgrounds, different ethnicities, male and female. And so at some point we wondered, well, what could be why is this recurrent mutation associated with such diverse phenotypes? Is it because it's sort of a hybrid? It's a overlap syndrome. Sometimes it's long QT, sometimes it's CPVT, sometimes we don't know. So we thought the best way to approach this would be to make a mouse. So we eventually made not one, but 2 mice, same mutation at the amino acid level, but in the different genes, the 2 different calmodulin genes, CALM1 and CALM2, now in the mouse. And of course, we used what is now a standard approach using genome editing to engineer those mice. And then they were bred for many generations to make sure we didn't track any off-target edits in the mice. And then just a little bit about the phenotype of both mice. We've just published this a month or so ago. Both mice had a relative bradycardia. Mouse hearts beat very fast, around 600 and something beats a minute. These mice all had relative bradycardia compared to wild-type mice. And they also had much greater heart rate variability. I think this is an illustration of that. They did have normal QT intervals under baseline. Now the QT interval is tricky to measure in a mouse, and I'm not going to stand up here and say we absolutely, did it exactly right. It's very tricky, and we did our best, but we didn't see any differences. And that said, when they get a slug of epinephrine, their QT is long. So there's a catecholamine-induced QT prolongation. The other thing that happens when you give them a slug of epinephrine plus caffeine to open the ryanodine receptor, in wild-type mice, you see tachycardia, as you would expect from giving a catecholamine challenge. But that's it. In the heterozygous mice, what we saw consistently with acutely, we saw tachycardia, and then that rapidly degenerated into ventricular tachycardia or ventricular fibrillation. And so it turns out this is just one of many types of ventricular arrhythmias we observed in these mice when they were challenged with epinephrine and caffeine. We saw everything from bidirectional ventricular tachycardia, which is commonly seen in catecholaminergic induced arrhythmias, like CPVT. We saw bigeminy, and of course we saw VFib and polymorphic VT. This was more prevalent in male mice carrying the CALM1 mutation, and least severe, least prevalent in female mice with the CALM2 mutation. The basis for that sexual dimorphism, we don't know. And the reason why maybe CALM1 is more severe than CALM2, we think is related to the fact that CALM1's transcript is at higher abundance in the heart of the mouse than CALM2. So it may simply be there is a higher mutation burden with CALM1. They're both severe, but CALM1's worse than CALM2, and males are more severe than females. We also observed, I don't show the data, but we also showed that male CALM1 mutant mice are not born in a Mendelian ratio. So and in fact, we looked intrauterine, and there are fewer mice that survive gestation with CALM1 males. So there was a survival disadvantage and embryonic lethality of males with the CALM1 mutation, not in females and not CALM2. I think this is just some other invoked arrhythmias that sort of make the point of CALM1 being worse than CALM2 and females being less severe than males. And then I think this is now, so the last thing on this story is, given that we think there's enough evidence to implicate a leaky ryanodine receptor in this disease, that we turned to an emerging therapy of CPVT, which uses flecainide, a well-known sodium channel blocker, to suppress that calcium leakiness of the ryanodine receptor. And this is work that was really done elegantly by Bjorn Nolman, who showed very nicely that flecainide wasn't having its efficacy in CPVT mouse models by blocking the sodium channel. Rather, it was by blocking the ryanodine receptor. Well, we thought, great, we have a cure for calmodulopathy. Well, not so fast. Flecainide did not prevent the arrhythmias invoked in these mice. So here are the arrhythmias, sustained and non-sustained arrhythmias in the mutant males treated with flecainide after a vehicle or flecainide, and there was no difference, same in the females. So flecainide was ineffective at treating this disease. that also gave us a little perspective on if this calmodulinopathy is also catecholamine induced, what similarities does it bear with CPVT, a very well-studied congenital or genetic arrhythmia syndrome? And in our opinion, they're not that similar. So here are the mouse models that have been published on CPVT. I previously had mentioned the calsequestrin knockout mouse, but there are also ryanine receptor mutant mice that have been studied. And all of these share, that, of course, they all have catecholamine-induced arrhythmias of different types. They all have increased spontaneous calcium release in the heart, and flecainide, at least in 2 of the models, suppressed the arrhythmia. But in our 2 calmodulin mice, and notice they were all on the more or less the same background strain. We saw, yes, they have catecholamine-inducible arrhythmia, similar dispersion of arrhythmia types, but in our case, and I don't show the data, we did look at spontaneous calcium release and did not show that there was spontaneous premature calcium release in these mice. There was blunted amplitude of calcium release in cardiomyocytes, which we think is probably a byproduct of low sarcoplasmic reticulum calcium load, possibly because the calcium has been leaking out and you've lowered the load. And then lastly, the bigger difference is that flecainide completely ineffective in this condition. So, now, the last thing I want to share with you is a very perplexing case that we're still trying to figure out. And this was a call I got related to a call I got from a pediatric neurologist in Chicago who said, literally, "Hi, how you doing? What do you know about calmodulin?" It's like, "What do you want to know? And why are you asking?" He said, "Well, I've been taking care of this boy, now he's 5 years old, who had what they considered to be kind of a benign seizure disorder early in childhood, started when he was about 2 years old, I think. Briefly treated with anti-seizure drugs, and he got better, and they took him off the drugs, and he wasn't developing right. So they thought, we're missing something. Let's send a genetic test. So they sent a whole exome sequencing test. And the only thing on the report was a pathogenic mutation in calmodulin. So my response was, okay, what's his ECG look like? So we didn't get one. But you better get one. Should you get it yesterday? So anyway, they referred him to a pediatric cardiologist. This is the position of the mutation, novel mutation, affecting, again, one of these critical calcium binding acidic residues in calcium EFHAN3. And we measured its calcium binding affinity. It was impaired. Nothing new here. What was new is he was not having arrhythmias. This is his ECG. It's not totally normal. He had kind of a borderline QT prolongation. His T waves are a little funny looking. And when he got epinephrine as a stress test, he did not get arrhythmias, and his QT didn't change very much. So he had a loop recorder put in, and for 6 months he was monitored closely, never once had an arrhythmia. But he started having seizures again, and his seizures occurred at a time when he was in sinus rhythm, sinus tachycardia. So anyway, we thought, okay, something's not right. Maybe this mutation is not pathogenic. Maybe it will not affect calcium channel gating, or it won't do something to the ryanine receptor. So we turned to our iPS cell-derived cardiomyocyte model. We transfected healthy myocytes with the wild-type calmodulin and compared it to this novel mutant calmodulin. And it looked like we lost calcium-dependent inactivation. Here they are lined up next to each other. Same stories I showed before. Wild-type calmodulin is associated with, and again, this is black line is calcium as a charge carrier, purple is barium. This difference is the degree of calcium-dependent inactivation, totally lost or nearly completely lost when we express this mutant. Okay, I don't understand what's going on here. Maybe this overexpression system is flawed. We're putting in too much of the mutant. Maybe the stoichiometry is bad. How about we get iPS cells from the patient? So we did. We grew those cells, we made cardiomyocytes, and lo and behold, the calcium channel kinetics are completely normal, not affected. So our next thought was, well, maybe there's allelic imbalance. Sometimes genes, different alleles are expressed at different levels. So we did quantitative RT-PCR multiple times and could never show a difference between expression of the wild type messenger RNA and the mutant messenger RNA. So that wasn't the explanation. But here is just sort of the summary of things. When we overexpress it, we see this impairment in calcium-dependent inactivation, but the native cardiomyocytes from the patient have normal calcium kinetics. So we're still at a loss. We're working on, we now have a series of cell lines from the patient for which we've corrected the mutation back to wild type to see if there's any difference. We've also taken the healthy control from this experiment and introduced the mutation. So stay tuned. We're hopefully going to figure this out. But so far, we have no clue why this. But it's important to know because perhaps this is an outlier for two reasons. One is not having arrhythmias. Possibly there is a protective factor. If we can identify that, maybe that's a handle on what we could target therapeutically. The second thing is, and we don't know the answer to this, he presented with seizures. Are those a primary manifestation of calmodulopathy, or he just has the common form of benign childhood seizures? We don't know the answer to that. So anyway, so just a brief summary here. I hope I got the message across that calmodulin, the 3 calmodulin genes, mutations or pathogenic variants in any of the 3 can cause a life-threatening genetic arrhythmia syndrome that we've nicknamed and others have named calmodulopathy. The features resemble, but are not exactly the same as congenital long QT syndrome or CPVT or a variety of others. There's often a lot of intermixing of features. We think the CPVT mechanism is mediated by impaired regulation of the ryanine receptor to allow the ryanine receptor to be too leaky, whereas the long QT manifestation is probably a defect in impaired calcium-dependent inactivation of the L-type calcium channel. And then our mouse models have been helpful to sort of investigate the more holistic side of things. And that's kind of a boring summary. So in this era of generative AI, I thought I would ask ChatGPT to write us a limerick. So here we go. Calmodulin in the heart has a role. Without it, the rhythm takes a toll. Heartbeat becomes erratic, a condition quite traumatic. It's important for keeping the heart whole. So anyway, with that, I want to thank the people who did the work, especially Lisa Wren, who did all the mouse work. Thank you.
As a precursor for CPVT, is that a direct effect of the mutation, or is that a potentially an indirect consequence of cytosolic calcium overload that's ultimately causing from the cytosolic side the ryanodine to be leaky, or is it really a manifestation of sodium calcium exchange manifesting as calcium waves? And have you looked at the exchanger?
Yeah, those are good questions. So the first question is this leakiness of the ryanodine receptor as evidenced by the increased frequency of spontaneous calcium waves in the myocytes, is that a direct effect of the mutant calmodulin? The evidence supporting that is simply that, and again, I didn't go over the data in that original paper with Bjorn Nolman as the senior author. Don Burr's lab actually measured calmodulin kinase activity, totally normal between the two conditions. So he ruled that out. And it's possible other mutations disrupt calmodulin kinase. We didn't see it in ours. Also, what was measured is calmodulin binding to the ryanodine receptor. Turns out it can bind more tightly, but it doesn't evoke the regulation. So we think there's something aberrant about the way calmodulin, which normally suppresses calcium release by the ryanodine receptor, is binding, maybe occluding other calmodulins from binding, but the mutant can't transduce its normal effect to suppress calcium release. The last question I think is really important, and I don't have an answer. Is this somehow due to aberrant sodium calcium exchange? And we have not looked at that. So, great question.
Wonderful talk and wonderful work. Thank you, Al. So you partly answered it just now, I think, sort of a mechanistic question and one very brief philosophical question. So the mechanistic one was going to be, I was trying to wrap my mind a little bit around the dominant negative mechanism and sort of where that comes from. I mean, you have 6 copies of the gene. you have 1 out of 6 that's abnormal. And how does that work? You should have plenty of normal calmodulin around, but you may have just answered that saying that if the mutant, because I thought you would have worse binding to calcium calmodulin dependent channels or other proteins if you don't bind calcium. But you said in some cases there's higher binding, but less functionality, less function. That may, I suppose that might be the answer.
Yeah, and I think the other way to look at it is, and I may have said dominant negative, it's definitely dominant. I think these, with regard to the calcium channel defect, I think of that as a dominant positive, because you've essentially got gain of function of the calcium. And you don't need many calcium channels to have that bad behavior to distort the action potential duration. And again, it's probably happening intermittently. And I think this is more of a dominant gain in the case of the calcium channel. Ryanine receptor, different. I think it is somehow impaired regulation. And so, and because it may be occluding binding of normal calmodulins, maybe it is a dominant negative. It's blocking the effect.
So my philosophical question is sort of a silly one, maybe to a geneticist, but these are obviously catastrophic clinical scenarios with anything that leads to sudden cardiac death. It's not treated on time. And so as more and more of these pathogenic catastrophic mutations that are completely absent in parents, de novo mutations, do you think as a clinical geneticist that everyone should have a scorecard? Should we be looking at these? Should we be looking for these in everyone from, in some registry fashion from people like a Danish registry when they're born and they get a scorecard?
Right. So prenatal testing, I think, is the question.
Basically,
Should we be
even
testing
more,
prenatally? And I think that question has been asked about many, dangerous mutations in different diseases. And I think it comes down to almost a financial question. Is it worth it? And certainly it's worth it for the family that lost a child. Is it worth it at a population level? And I'm not the person to ask about that. So I think it's an implementation question, as it turns out.
Has there any been assessment of this patient's SID populations? I'm curious.
That's a great question. The question was, if people couldn't hear it, is there a connection with these gene mutations and sudden infant death syndrome? It's been looked at. It's not overt. There's been a lot of interest, including some work that we did years ago, collaboratively with groups that had databases and DNA registries of SIDS cases. And about 15% or so of SIDS, what's diagnosed as SIDS, is probably undiagnosed congenital arrhythmias. Calmodulin's probably not factoring into that very much. And I don't know why that is. It may just simply be ascertainment bias. So.
Very nice talk. I really enjoyed it. So one clinical question and 2 basic science questions. One is that have you ever tried sympathectomy in your patients as it is kind of this catecholaminergic rhythm? And as probably like CPVT, they get a storm and AICD is not actually very effective. The basic is that 2-part. One is that have you looked at the FK binding protein as an inhibitor of ryanodine binding to the ryanodine receptor as a result of these mutations? One, and then you had also.
I better answer those before I forget all of them. So the first question is a really good one. Has sympathectomy been effective clinically? This International Calmodulin Registry collected a lot of data on that. It doesn't seem to be a transformative benefit. In fact, the first case I presented, that child by the age of 9 months, I think had bilateral sympathetic denervation, and it didn't seem to help. Your second question, I don't have an answer for. Does FKBP influence the ryanodine receptor differently in the presence of these calmodulin mutations? We don't have an answer for that.
And then you mentioned one of these patients had some glucose abnormality, and you kind of attributed that to the propranolol. My question is that actually calcium calmodulin is very important for insulin secretion in the pancreas. could that be the defect in the beta cell?
Yeah, it's a great question, and it was considered. I think we don't, obviously, because the child was deceased, we could only sort of speculate about that. But the patient was treated with propranolol, which does have, as a known side effect, hypoglycemia. I'm not sure I know the mechanism, but what you brought up was important, whether or not the defect in the calcium channel that we believe is responsible for the arrhythmia may impair insulin secretion. And I don't know the answer to that. Kind of conceptually, maybe. And I think there's been evidence. So in a different calcium channelopathy called Timothy syndrome, which has mutations in the calcium channel, they're also susceptible to hypoglycemia. And one wonders if, in fact, there may be a mechanism of hyperinsulin secretion. I don't know that that's been measured. So I can't really give you a definitive answer. So, possible.
Not to experience sudden death, do they, live a fairly normal life? Do they have ejection fraction issues? Having a lot of cytosolic calcium overload throughout.
Yeah, it's a great question for those individuals who are genotype positive. In other words, they have a CalModulin mutation, but are not dying during infancy or having cardiac arrest. What's their longevity? I think it's too early to know. I mean, this was really kind of 11-year-old story so far. So the registry that I mentioned does have people into their 20s and maybe 30s. And I know one of the index cases that we studied was at the time we studied, the family was in her 20s, but she had had CPVT early in life and was chronically on beta blockers and was told not to exert too much. So I think there may be a subgroup that does survive. And then there are these families for which they transmit the mutation from parent to offspring that seem less severe. And part of that is these are induced arrhythmias, and there has to be a scenario to induce the arrhythmia. And if they're inactive or are less subject to that, maybe they survive. We have, I think of these severe mutations, they've all been de novo. And as far as we can tell, there has yet to be a severe mutation passed on to another generation. But I think it's still maybe premature to say much about that since we don't have all the data. Anyway, okay, right on time. Thank you.
Last time meeting.
Do I need to do anything here? It says I'm just going to hit.